Good afternoon, student. So today we are going to read about the microbiology of water. In the last class, we read about the microbiology of land or the soil. And in this lecture, I'll tell you about what all kind of microbes which are present in the water, what is their implication, are the pathogens, how to control them, and all those things we need to know. So I'll share the PPT with you and keep explaining this presentation about the microbiology of water, and at last I'll share this recording as well as the PPT with you. All right, so share the screen. All right, so microbiology of the water. So first of all, let's read about some of the important facts about the water. So as you all know that 70% of the earth is water, and out of that all water, 97% of the total water is in oceans, and 3% is a fresh water in the form of rivers, lakes, glaciers. So in the 3%, 2% is in the form of glaciers or not available for drinking or other purposes for the use, and the 1% is the available water which is in the rivers and lakes. So you can understand how less amount of water which is available to us for our drinking or potable water drinking purpose or any other use. And this is like going to be a big scarcity in future, because if we see that number of people with water shortage are almost 0.5 billion now. Right now the population of the earth is almost 7 million. Out of that is 0.5%, 0.5 billion is have a water shortage, and this is estimated by 2025 this will be almost 2.8 billion people which will be have the water shortage. So you can understand how big this whole scenario is, and how it is important to secure the water, to do not waste the water, as well as to keep the water cleaning. And in this, microbiology plays a very huge role, very big role, because number of microbes are present in the water, and those could be beneficial microbes as well as the harmful microbes. So we need to control those harmful microbes or the pathogens in the water. We don't need to pollute the water and all those things. So in this row there are terminology which are associated with the water, like the potable water, you know, that clean water, free from all objectionable material. All objectionable material means including pathogen, taste, odor, color, toxin, radioactive material, organism, oil, gases, petroleum, etc. So it should be free for everything, means it is a drinkable water, almost drinkable water, that is potable water. Then comes the fresh water. So that 1%, which what we are talking about, that 1% is fresh water, which means that is a non-salt water and available as a fresh water. Out of that, how much is potable depends on how much is a contaminated, and remaining is can be used as a potable water. And the pollution, anything that makes it non-potable, non-drinkable is a pollution. And sewage, the community waste or garbage we dump into the sewers or which will ultimately go to the rivers. So we already know now that the water scarcity is going to be the big challenge in the near future. Still, what we do, we waste a lot of water, and as well as we contaminate a lot of water with the community waste and everything. So that all needs to be taken care. In our last lecture about the wastewater treatment, that was our aim, to how to clear the water so that that water can be reused again and again and can be regenerated again and again, so that our groundwater level should not decrease, water remain surplus at all the places, at least not surplus, but suffice the need of the community, suffice the need of our society. And all this thing can be done when we control the pollution and we understand about the sewage, waste and garbage that need not to be dumped into the sewers, which will ultimately go to the rivers. So what about microbes in the aqueous environment or in the water? So ecology of aquatic environment is very complex because there are a number of types of microbes, pathogens, viruses, bacteria, and or as well as the protozoans or the parasites which are present in the aquatic complex. In fact, the first life was originated in the water. So actually microbes were first actually originated in the water and then they came to the land. So there is no question that if that the microbes about the presence of microbes in the aquatic environment. And microbes has evolved to live in saturated salt solution, no problem. Like even if it is a very high salt lake like the Sambar Lake, you can still find number of microbes in the Sambar Lake. They can grow at freezing point, below freezing point, as well as up to the 110 degrees centigrade. Water they can grow in water full of toxins like the copper, cyanide, lead, silver, gasoline, oil, benzene, and other things. So they can grow in the polluted waters. And coliforms are the type of bacteria which are non-spore forming gram negative bacteria, and which are actually uh, which can ferment lactose with the production of acid or gas, and they are generally present in our alimentary canal. So coliforms are actually widely present in the um, in the water. Generally, they are not pathogens. I'll define it later in this presentation, and why coliforms are important when we read about the microbiology of water. So next is water quality standards on the basis of microbiology. So water quality standards can be on the basis of different things, on the basis of pollutants, on the basis of heavy metals, on the basis of microbiology. So we are reading about the water quality standard on the basis of microbiology. So drinking water, no coliform contamination acceptable. So there should not be any coliforms, any bacteria which should be in the drinking water. Recreational waters, 200 fecal coliforms per 100 ml is a limit for the recreational water. What is recreational water? The water in the swimming pools, water on the beaches. Then the fish and wildlife habitat, where the wildlife habitat and the fish like the lakes are there. The acceptable limit is like the 5,000 fecal coliforms per 100 ml. And if there are 14 fecal coliforms per 100 ml, then we can say that there could be a contamination of the shellfish in the water. So these are different water quality standards. For drinking water, I can tell you there is no coliform contamination is acceptable. So detection, generally how we detect these coliforms, generally by the most probable number. In our one of our lecture, we already read about most probable number, how we do it. If you remember, I told you that you take the samples and the tubes with the medium, 9 ml medium in tubes, and then we do the serial dilution up to the tube last tube where you get the growth, and then you back calculate with the table, with the refer with the table to determine the statistical range of number of bacteria or number of coliforms. So you understand? I hope you remember that how we calculate the most probable number. We use 10 ml of the sample, 1 ml or 0.1 ml of the sample, whatever range you want to use, depend on how many bacteria is there, and then you do the serial dilution till the tube you dilute it, you back calculate how much bacteria is there present there. So that is a kind of detection by the most probable number. This is generally used in the industry to calculate the number of coliforms. And how they determine, not just by the growth of coliform, but they also in the medium which they take is a lactose medium. And in the lactose medium, what coliform do? Coliform reduces the lactose and produce gases and acids. Does not. So what is a limitation of the most probable number? Does not detect the total number of bacteria, as well as cannot tell you about the specific pathogens, but still it gives you an idea that there are microbes present in the water or not. So pathogens are the most concern in fresh water. So fresh water generally can have a number of contamination of bacteria, viruses, and parasites like the protozoa. So these are the different bacteria which can have the fresh water can be contaminated by these different bacteria like Salmonella, Shigella, E. coli, Campylobacter, Yersinia enterocolitica, Staphylococcus aureus, Bacillus cereus, Clostridium species, different kind of Clostridium species, and the Vibrio species. All these bacteria are, which are named here, are pathogens. So these are pathogenic bacteria which are generally can be present in the fresh water, and this is very dangerous. And viruses, with like the different kind of viruses which can cause hepatitis A or the Norwalk disease. And then the last one is the protozoa or the parasites like the Giardia, Entamoeba, Toxoplasma, Sarcystis, Isophora, Cryptosporidium, Amaria, and Cyclospora. All these are protozoans which can again cause the number of diseases. We'll read about them one by one. Right. So these are the bacteria generally found on the fresh water, on the surface water. These are the different type of bacteria. This is a disease which they cause, and these are the infections of the infections caused by the the symptoms of the disease. So Aeromonas, it causes enteritis, and the symptom is very thin blood and mucus containing diarrhea. Campylobacter zooxanthellae is the disease which is causes Campylobacteriosis, and it is flu, diarrhea. So in most of these diseases, you should understand that diarrhea is one of the main problem. So as soon as you drink water, and if there is a diarrhea, the first and foremost thing is check your water. There could be a problem of pathogens in the water. Flu, diarrhea, head and stomach aches, fevers, cramps, and nausea. Then E. coli, urinary tract infection, neonatal meningitis, and intestinal diseases. So watery diarrhea, headaches, fever, homolactic uremia. What is homolactic uremia? Homolactic uremia is that uremia is presence of urea in the blood, and homolactic uremia is that this presence of urea in the blood is from any of the water which is generally used in the home. So it is homolactic uremia. So the disease has come from the home water which is used, and it is the presence of urea in the blood. And then the kidney damage. Then next is Plesiomonas shigelloides in the diseases, Plesiomonas infection, and nausea, stomach aches, watery diarrhea, and sometimes fever, headache, and vomiting. All these are the symptoms of this disease. Then comes the Salmonella or typhus. It is the disease is typhoid fever, and it is fever. Symptoms is fever. Salmonella, another disease, salmonellosis, sickness, intestinal problems, vomiting, diarrhea, and sometimes slight fevers. Then comes the Streptococcus. It is again intestinal disease, gastrointestinal disease, stomachache, diarrhea, fever, and vomiting. And then the Vibrio altor. This is generally present in the fresh water. It is a light form of cholera, and it is the symptoms is diarrhea. Vibrio also cause another disease which is known as what? Cholera. So the organism which cause this disease is Vibrio cholerae, and cholera is one of the important disease to know. It is caused by Vibrio cholerae. Initially, it was very high in 1800. It is very prevalent in US, but now it has been mostly controlled in US and Western countries because of the hygienic water. And currently, Asia, Africa, and Latin America, still there are a number of cases of cholera in the Asia, Africa, and Latin America because the hygienic condition, water hygiene conditions are not so great. And about 95,000 to 1 lakh deaths per year from this disease, cholera, throughout the world, and transmitted through water, fresh vegetables, and the shellfish. Then the disease which are caused by the protozoa. So we have read about the bacteria. Now the diseases which are caused by the protozoa. So amoeba or Entamoeba histolytica. The disease is amoebic dysentery, severe diarrhea, headache, abdominal pain, chills, fever. If not treated, can cause liver abscesses, vowel perforation and death. So Entamoeba histolytica does this, and this is very dangerous. Then Cryptosporidium parvum, the disease is cryptosporidiosis, feeling of sickness, watery diarrhea, vomiting, and lack of appetite. Then next is Giardia, the disease is giardiasis, diarrhea, abdominal cramps, flatulence, belching, and fatigue are the symptoms. Flatulence is like the formation of a gas in the stomach, and belching is feeling like a fire in the stomach. So that is belching. Okay. So flatulence and belching, these all are the generally with the diarrhea. You also have these two related. Then next is Toxoplasma gondii. The disease is toxoplasmosis, flu, swelling of lymph nodes with pregnant women, subtle abortion, and brain infection. So this is very harmful if it happens to a pregnant woman, because it can make abortion and the brain infection. All right. So we have read about here is a Giardia and Cryptosporidium. So gradiasis and cryptosporidiosis, these are the disease which are very severe. Both are by protozoans. Transmission through water, 97% of all surface water carry cyst for this. So you can understand that all that 1% of the fresh water have these cyst of these bacteria, these protozoans, and this can grow. Resistant to chlorine but can be filtered. Okay, so that is an important thing. If because these are cysts, they are resistant to chlorine. So if you do a chlorine treatment to a water, probably this will not kill. But if you filter it with the 0.2 micron filter, this can be filtered. So generally the filters which are coming for the home have those filters which can filter this giardiasis and cryptosporidiosis with giardia and cryptosporidia, so that we don't get these diseases. One of the outbreak in 1993, known as Milwaukee outbreak, it killed all 1 lakh people just because of this, disease. And at that time, it was ordered that almost like 7,000 beaches all around the world was closed just because of this disease. And this was a very important, very important in history, because this has, at that time, it took a long time to find out that why this disease is, the people are getting this disease, and then they come to know that these are due to giardiasis and the cryptosporidiosis. All right, so public and water supply. So whenever the water is supplied to the routine monitoring of water quality is done with the indicator organism. So there are a number of indicator organisms which are present in the water. And if these organisms are present, probably we should also can conclude that there are pathogens which are also present. So these indicator organisms are used to know that the water is of potable quality, or water cannot be used as a potable water. So this indicator organisms are generally the coliforms, different coliforms. To determine if fecal coliforms are from human or other animal, must test for the fecal streptococci. So that is very important. What happened? There are fecal coliforms are there, and then the fecal streptococci are there. And for both of them, there are different tests. Fecal coliforms are different tests done for fecal coliforms, and different tests is done for fecal streptococci, or we see that the growth on the plates, number of colonies. So the ratio of fecal coliform upon fecal streptococci will tell you that probably from which organism they are coming, like from human or from any other animal. So we can know the source of the contamination by finding the ratio of fecal coliforms upon fecal streptococci. So for example, if the ratio is near about 4.4 fecal coliform upon fecal streptococci, then we can be very sure that that it is probably from the human, because if the ratio is fecal 4.4 means the fecal coliforms are higher than the fecal streptococci. For the duck, if the contamination is coming from the ducks, like because ducks are always in the river, and if this water is potable water is taken from the rivers, then it is 0.6. For sheep, for chicken, and for pig, if the contamination are coming from these three, then the ratio is 0.4. If it is coming from cow, the ratio is 0.2, and if it is coming from turkey, the ratio is 0.1. So you understand that this all ratio can tell you the probable origin of the contamination in the potable water, from where it is coming. So this ratio, you don't have to learn this ratio, but you just understand that if fecal coliforms are in the human, fecal coliforms are higher than the fecal streptococci. And if this ratio is if your contamination from the water has this ratio, then probably the contamination is coming from water. And if it has this ratio, probably the contamination is coming from turkey. Got it? However, this is not always true. I'll tell you why. But first, let's read the characteristic of a useful indicator. So indicator organism, I told you, are the coliforms. But what, so coliform are the examples, okay, for the useful indicator. What all characteristics should be there in the useful indicator? Useful for all water types, always present when pathogens are present. So that is the most important character, that the coliform should be present when the pathogenic bacteria or anything any other pathogen is present. Not present in the absence of pathogen. That should be the another characteristic, that it should not be present in the when the pathogen is absent. Suppose if it is not present when the pathogen is present, and if it is present when the pathogen is not present, then there is no that is not a good indicator. So it should be present. You should give a positive test when the pathogen is present. You should give a negative test when the pathogen is not present. Correlate with degree of pollution. If that number is high, you can say that the contamination is very high. If the number is low, you can say that the contamination is low. More easily detectable than a pathogen. It should be directly detectable by some kind of test, microbial test, like for coliform we do a lactose test, something like that. So it can be done easily by the easily detectable test. Survival longer than the pathogen, and it should survive in that water for longer than the pathogen. Suppose if it dies before the pathogen and the test is coming negative, then we will not be sure that the pathogen is present or not. So its life cycle should be higher than the life cycle of a pathogen, and known dangerous to work with. And if the microbiologist is working with this indicator, microbial indicator, like the coliform, it should not be any dangerous for that microbiologist. All right. So yeah, so these are the common bacteria, common groups of indicators, like the coliforms, streptococci, and the spore fermenters. So for the coliforms, the tests which are done is for total coliforms, fecal coliform, or the E. coli. Whereas the streptococci, fecal streptococci and enterococci are the indicators, and in the spore formers, Clostridium perfringens is used as a indicator. So general indicating organism are indicator organisms like our general coliforms, which are found everywhere, and then the fecal coliforms from the mammals and the bird species. So these are fecal coliforms, and then the Enterococcus bacteria. These are type of fecal streptococci from the species of warm-blooded animals. So these are the coliforms. They generally don't make people sick, so they are not pathogens. But presence of them showed that they are pathogens which are present in the water. So presence of them make the water not drinkable. Although these are not the pathogen, but their indications is a indication of presence of pathogens in the water. So they are strictly no for the drinking water, drinking quality water. Then next comes the there are problems, there are drawbacks of the coliform indicator test. And what are just drawbacks like false positive, like the Enterobacter aerogenes. It gives a false positive for the coliform indicator test, as this is a non-pathogenic bacteria. But if it is present, that coliform test is positive, although this is not an indication of a pathogenesis. But if this is present, the coliform indicator test is positive, which means the water we use is indicated as not drinkable. However, this is not a pathogenic thing. Similarly, false negative, like the Salmonella typhi. You know that Salmonella typhi causes typhoid, but this presence of Salmonella typhi gives the coliform indicator test a negative. So although coliforms are not present, but Salmonella typhi is present, this pathogen is present, which can cause typhoid. So this is like a false negative. And then next, drawbacks of coliform indicator test. Other drawbacks is like as I told you, that there's a ratio between the coliforms and the streptococci, and this ratio can indicate that from where the contamination is coming, from human, from birds, from other animals. But this ratio can also change because of the infection which is present in the population. So if there is an infection which is present in the population, this feces or this contamination is coming to water, probably this ratio will change, and we'll think that this water is coming from any other animal. However, it could be from the human population. So that is another drawback of the coliform indicator test. Another is that if the population is taking any treatment, or if there are environmental factors which can also change this ratio. So these are the things. Third is chlorination. When we do the chlorination of the water, the pathogens are killed, and it becomes a indicator that the pathogens are not present. However, there could be lots of viruses which are present, which can cause number of diseases, as well as chlorination does not kill protozoans or parasites, and that could be the reason of another disease. So, and another is pathogen can hide this coliform test by sticking themselves to the suspended solids. So although these pathogens are present, but the coliform test is negative. So although this test has a number of drawbacks, but still it is used by the industry which are dealing with the water, like the cold drink industry or the portable drinking water, packaged drinking water industries. They all have to compulsorily do the coliform indicator test. Then there could be other tests which can be done for the pathogens, like the direct test for pathogens involves cultivation method or the molecular test. Cultivation method, we'll cultivate the pathogen and see that whether the pathogens are present on the different medium. However, this is time consuming, expensive, and there could be potential dangers to the lab personnel, because we are growing pathogens. So we need a BSL-3 biosafety level 3 labs, as well as the lab personnel it could be dangerous for the lab personnel. Second is molecular tests, directly testing the DNA, directly getting the DNA sequence and finding the pathogen. It is expensive and it requires a huge expertise. Then in addition to the bacteria and protozoans, there are a number of viruses which can be a cause of disease, waterborne diseases, like the hepatitis A, inflammation and necrosis of liver. Then the Norwalk type virus, which can cause acute gastroenteritis. Then the rotaviruses, acute gastroenteritis, and especially in children. Enterovirus, many types affect intestine and upper respiratory tract, and reovirus infects intestine and upper respiratory tract. So you know that number of virus diseases can also be caused because of the water contamination. And nowadays there's one of the virus which is actually not a waterborne, not even airborne, but it caused number of and the problems, that corona SARS-CoV-2 virus. Although this is not it is not airborne or it's a waterborne, but still it remains on the surfaces, number of surfaces for a longer duration, that it is a cause of contamination and cross infection, and you can see that how it is impacting the world. So virus detection can be done by a number of technologies like electron microscopy, immunoassays, cell cultures, and the most important, reverse transcription polymerase chain reaction. So for SARS-CoV-2, what is the detection kits have reverse transcription polymerase chain reaction. So what we do in this, this is a RNA virus. So we initially convert RNA to DNA by reverse transcription, and then this DNA is quantified by polymerase chain reaction. So what we are doing for the viral dissection for the SARS-CoV-2 is RT-PCR, and this is generally done for number of viral detections. Other tests could be electron microscopy, immunoassays, and cell culture, but these all are again they take more time than the RT-PCR, as well as immunoassays are not very successful because they can only be used when the pathogen is able to activate the antibodies in the active antibodies in the body. So until unless there's a antigen antibody reaction, the immunoassays are not successful, right? And the next thing is chlorination of the water. So this is very important. So next thing is chlorination of water, and in the chlorination is done by all the public water department. We supply the water, potable water, to the communities, to the societies, because chlorination is very effective where other treatments cannot be done. So in the chlorination, there are two methods of chlorination. Shock chlorination, where the 50 to 100 ppm of the dose is contacted with the water for 6 hours. That is shock chlorination, kill all the pathogens which are present in the water at that time. And then there is a continuous chlorination. A small dose of chlorine is continuously pushed with the water in the water, so that a measurable amount of free residual chlorine remains there. And this free residual chlorine is actually important for killing any kind of pathogens, or for to stopping the growth of any kind of pathogen in the water. So what is this free residual chlorine? So this can be explained with the help of chlorine doses. Chlorine doses actually the total chlorine which is added, and then the chlorine demand, which is a chlorine which is in the form of inorganic salts, and then the combined residual chlorine is a chlorine which has been reacted with the organic material which is present in the water, and then last remaining is free residual chlorine, which is the active chlorine which can kill the pathogens which are present in the uh, water. So suppose this is whole is a chlorine dose. This part is chlorine demand, which is like a inorganic chlorine, and then the rest, this is a combined residual chlorine, which is a chlorine which has been combined with the organic material which is present in the water, and then this rest is a free residual chlorine which is remain for the bacteria. So this is inorganic chlorine, chlorine demand. Combined residual chlorine is organic, and the free residual chlorine is responsible for killing or for stopping the any kind of growth of any kind of pathogen in the water. So this free residual chlorine, actually what is this? It kills the bacteria, and the bacteria kill rate is proportional to the concentration of free residual chlorine. And this can be tested in the with the number of kits which are nowadays available in the market. Like this is autochlor, which can say that how much free residual chlorine is present in the water. All right, so this is all about the water microbiology, and this is your last lecture. I'll share the PPT and this video with you. Kindly read them, and we'll talk about the assignment on email, or about the test and the assignments. Thank you.